Melatonin membrane receptors have been identified in the SCN of vertebrates. The signal transduction pathways seem to involve both MT1 and MT2 receptors, inducing an increase in the expression of two clock genes, period 1 and period 2. Without the feedback information from melatonin, the SCN would not properly interpret natural photoperiodic changes, exhibiting a free-running internal rhythm with a cycle longer than 24 hours. This phenomenon is apparent in completely blind animals and humans whose eyes, specifically the MRGC, do not appropriately receive environmental photoperiodic information. Importantly, administering melatonin to blind subjects partially re-entrains their biological rhythms closer to normal, indicating the crucial role of melatonin in maintaining the circadian rhythm. The pineal gland mainly consists of pinealocytes, microglia, and astrocytes. Pinealocytes, differentiated from PAX6 expressing neuroepithelial cells, specialize in synthesizing and releasing melatonin, possibly along with other substances. Pinealocytes contain a significantly higher number of mitochondria compared to neuronal cells. These mitochondria exhibit dynamic alterations related to their fission, fusion, and mitophagy activities during a 24-hour period. Due to the high density of mitochondria, it is speculated that they are the primary sites for melatonin synthesis, a speculation confirmed by subsequent studies. Melatonin synthesis occurs in the matrix of mitochondria, although it can also occur extramitochondrially, albeit less efficiently due to substrate availability. During the night, when melatonin synthesis peaks, an increase in mitochondrial fusion is observed, while more fission is noted during the day. These dynamic changes in the mitochondria are believed to be associated with their function, primarily the production of melatonin. Recent studies have shown that melatonin itself can regulate mitochondrial morphology. It promotes mitochondrial fusion by upregulating the levels of mitochondrial fusion proteins like midophysin-1, melanophin-1, and ULPA-1 and inhibits the nuclear translocation of dynamin-related protein 1, DARP1, which increases mitochondrial fission. Consequently, melatonin promotes mitochondrial fusion and reduces mitochondrial fission. The effects of melatonin on mitophagy, the removal of damaged mitochondria, are still unclear. Some studies suggest that melatonin inhibits mitophagy, while others indicate that it promotes this process, depending on the experimental conditions and cell type. It remains uncertain whether the mitochondrial dynamic changes in pinealocytes are related to their functional activity controlled by clock genes or a result of their melatonin production rhythm. Apart from pinealocytes, astrocytes and microglia in the pineal gland also have the capacity to synthesize melatonin efficiently. It is hypothesized that melatonin synthesis is coordinated by both pinealocytes and macrophages, /glia and astrocytes, particularly for immune responses. For instance, during an acute inflammatory response, the transcription factor NFK switches melatonin synthesis from pinealocytes to macrophages/microglia and then back to pinealocytes upon resolution of the inflammation. This coordination enhances the pineal gland's capacity to generate melatonin, especially under conditions like oxidative stress or inflammation. The primary function of astrocytes and microglia in the pineal gland is to regulate pinealocyte melatonin synthesis under normal conditions. Various conditions, including elevated intracellular calcium concentration, can excite astrocytes/glia, resulting in NFKB activation. These excited cells then release soluble TNF-alpha, a signaling molecule that inhibits melatonin synthesis in pinealocytes by targeting AANAT. Other regulatory mechanisms, such as purinergic signaling, may also influence melatonin synthesis in the pineal gland. Once synthesized in the pineal gland, melatonin is rapidly released through several outlets. Traditionally, it was believed that melatonin is released into the precapillary spaces, then enters the capillaries, and reaches the general circulation through surrounding veins and sinuses. However, recent findings suggest a more significant route where melatonin is directly released into the cerebrospinal fluid of the third ventricle in mammals. This results in extremely high melatonin levels in the CSF of the pineal recess of the third ventricle, several orders of magnitude higher than in the blood. There are at least two parallel melatonin secretory routes: the general circulation and the CSF. A critical question arises regarding which route primarily transduces the photoperiodic information from the retinas to the SCN pineal loop, especially to the SCN. Given that CSF melatonin is not derived from the blood, but directly from the pineal gland, the role of blood melatonin circadian rhythm as the signal of natural photoperiodic information is questioned. It is hypothesized that CSF melatonin, rather than blood melatonin, serves as the signal of natural photoperiodic information. It is widely recognized that many foods contain melatonin, including herbs, vegetables, fruits, cereals, beans, eggs, meats, fish, milk, wine, beer, and coffee. Consuming these food items can increase circulating melatonin levels. In some cases, elevating serum melatonin levels to as high as the nighttime peak levels of melatonin. The impact of food-derived melatonin on signaling information and its potential chronobiological consequences remain unclear. If the melatonin in the general circulatory system serves as the photoperiodic signaling, food-derived melatonin might influence chronobiological effects. Conversely, if the cerebrospinal fluid melatonin serves as the exclusive signaling to the suprachiasmatic nucleus, SCN, the serum melatonin derived from food would not affect chronobiology, since it is unlikely to reach nighttime CSF melatonin levels. The SCN is likely regulated by the high levels and square shape of the melatonin rhythm, which is entirely different from the serum melatonin concerning their shapes. It would not respond to the low level and other shapes of melatonin rhythm input. Therefore, the photoperiod-induced melatonin message is a precise trait that would not be influenced by non-CSF melatonin level alterations. The pattern of the melatonin circadian rhythm in the CSF of the third ventricle is similar to that of the pineal gland, rather than in the plasma. This data was obtained from long-term pineal gland dialysis in a free-running rat. Extrapineal-generated melatonin and diet-derived melatonin might increase peripheral plasma melatonin levels, but they do not mimic the pattern and reach the high level of the melatonin circadian rhythm in the CSF of the third ventricle to impact the function of the biological clock. High levels of central nervous system melatonin exhibit protective effects on brain tissue. Pinealectomy, the removal of the pineal gland, results in accelerated neurodegenerative changes and signs of premature aging in animals. Pineal grafts in the brain protect it from oxidative damage induced by ischemia/reperfusion in mice. These effects are mainly attributed to the antioxidative, anti-inflammatory, and anti-apoptotic effects of CSF melatonin directly released by the pineal gland. Pineal calcification, known by various names including corpora arenacea, acervulae, brain sand, somoma bodies, and pineal concretions, was first observed in humans as early as 1653. This phenomenon is widespread across species including humans, ox, sheep, horse, donkey, monkey, cow, gerbil, rat, guinea pig, chicken, and turkey. While concretions were not found in the pineal organs of fish, amphibians, and reptiles, high calcium content was detected in their pineal glands through ultrastructural calcium histochemistry. The rate of pineal calcification increases with aging, and in some species, it reaches as high as 100% with age. Interestingly, calcification also occurs in neonatal humans. There are differing views on the implications of pineal calcification. Some researchers believe that it might be a physiological process not associated with pathological or aging changes, but rather related to the metabolic activity of the pineal gland itself. In gerbils, the accumulation of pineal calcium deposits is blocked by superior cervical ganglionectomy, which is believed to shut down the function of the pineal gland completely. Furthermore, gerbils exposed to short photoperiods exhibited significantly higher numbers of pineal concretions than those exposed to long photoperiods. Pineal calcification was enhanced in gerbils with bilateral optic enucleation, where the animals are completely devoid of photoperiodic influence, generating more melatonin. This evidence supports the metabolic theory of pineal calcification. Contrary to the metabolic theory, a substantial amount of evidence suggests that pineal calcification is indeed associated with human pathological disorders and aging. Several studies have pointed out the relationship between pineal calcification and schizophrenia, and the highest pineal calcium content was detected in patients who died of renal disease associated with hypertension, among other diseases. Recent studies have reported a strong association between PGC and neurodegenerative diseases, particularly Alzheimer's disease. This association is connected with the melatonin levels synthesized by the gland. Melatonin is well established as a neuroprotector due to its potent antioxidant function and anti-inflammatory activity. The brain, rich in lipids and lacking the antioxidative enzyme catalase, consumes a large quantity of oxygen, making it more vulnerable to oxidative stress than other organs. A decrease in endogenous melatonin results in neurons being less resistant to oxidative stress or brain inflammation. Several studies have reported a negative association between Alzheimer's disease and serum or CSF melatonin levels. Mechanistic investigations have revealed that melatonin inhibits the secretion and deposition of beta-amyloid protein and suppresses tau protein hyperphosphorylation, reducing intracellular neurofibrillary tangles, another biomarker of Alzheimer's disease. Animal studies have shown promising results regarding the use of melatonin in the treatment of Alzheimer's disease. Large doses of melatonin, precisely 100 milligrams per liter in drinking water or 10 milligrams per kilogram of body weight per day, have been found to prolong the lifespan of animals with single, double, or triple gene mutated Alzheimer's disease models. These doses also positively modulated biochemical and morphological alterations and improved cognitive performance. However, such large doses have not yet been applied in clinical trials for Alzheimer's disease. Notably, melatonin treatment in sporadic Alzheimer's disease animal models, OXYS rats, produced impressive results, particularly in preserving the microstructures of hippocampal neurons and their mitochondrial distribution and integrity. These observations provide solid evidence, suggesting the potential use of relatively large doses of melatonin in the clinical treatment of Alzheimer's disease. In a few cases, melatonin treatment did not yield expected results in Alzheimer's disease patients or in animal models. However, no serious adverse effects of the treatment were reported. Moreover, melatonin has also shown positive results in patients and animal models for other neurodegenerative diseases, including Parkinson's disease, amyotrophic lateral sclerosis, ALS, multiple sclerosis, MS, and Huntington's disease. For instance, in MS patients, melatonin supplementation improved biochemical markers and alleviated some symptoms. In most vertebrates, melatonin production decreases with aging. This decline can be attributed to two main factors: a dampened melatonin synthetic capacity due to reduced density of beta-adrenergic receptors in the pineal gland and downregulation of gene expression or phosphorylation of AANAT/SNAT, and increased consumption of melatonin due to metabolic alterations, such as higher OROS generation by aged cells. Consequently, melatonin is used more to neutralize the overproduced OROS in aging organisms, resulting in lower levels in aged vertebrates. Low melatonin levels are considered a biomarker of aging. Depressed melatonin production, either by pinealectomy or aging, accelerates the accumulation of oxidatively damaged products, hastening the aging process. Conversely, grafting young pineal glands to older animals or supplementing with exogenous melatonin significantly increased the lifespan of experimental animals. Recent advancements in technology, such as PET scans and susceptibility-weighted magnetic resonance imaging, have enabled the detection of even very small pineal concretions, which were previously undetectable. This has revealed that the rates of pineal calcification have been significantly underestimated in the past. Current data indicates a high incidence of pineal gland calcification across various countries, regions, and races, challenging the notion that PGC is a normal physiological process. Pineal gland calcification is often associated with decreased melatonin levels and several pathological alterations, including neurodegenerative diseases such as Alzheimer's and multiple sclerosis, migraine, symptomatic intracerebral hemorrhage, symptomatic cerebral infarction, sleep disorders, defective sense of direction, and pediatric primary brain tumor. Interestingly, PGC is mainly associated with brain-related disorders, but not with other organ pathophysiologies, even though decreased melatonin levels were detected in the blood, which supplies all tissues. This observation further supports the hypothesis that high levels of melatonin released directly into the cerebrospinal fluid from the pineal gland serve as the biological circadian rhythm regulator and the neuronal antioxidant, while the blood melatonin is the residue of the pineal melatonin. This residual melatonin only resembles the CSF melatonin rhythm and may be without significant biological functions.